everyone, welcome back to the cell signaling course. If you recollect, we were discussing receptor serine threonine kinase in animal kingdom. We also saw a lot of examples where signaling through receptor serine threonine kinase was playing very important role in infection, immunity, gonadal development, allergy response, cancer and so on. We also saw clear examples of receptor serine threonine kinase signaling mediated through SMAD-like molecule, unlike the MAP kinase pathway, which was the preferred pathway in plant kingdom. Now we are going to switch gear and start discussing a particular receptor kinase activity which is present only in the animal kingdom. And we are going to discuss today the receptor tyrosine kinases. But before we go and discuss the receptor tyrosine kinases, let's see what we have understood about in general in the signaling. The signaling could be very simple as a linear thing where ligand binds to the receptor. This receptor activates the effector, and this effector through its end result gives a response. And this is where each step amplifies the signaling. Here in case of signaling where linear flow of information happens, ligand binds to the receptor, activates effector and which mounts the response. At every step you have amplification of the signal. So you can understand if there was an intensity of response started with ligand was x, it becomes 10x, it becomes 100x, eventually a 1000x amplified response is seen. You also have a signaling where divergence happens, where ligand binding to the receptor is unaltered, but there are more than one effector molecule, where we saw in case of two component system, simple response regulator and a response regulator with additional domain carrying out different functionality. Now downstream of these effector molecule, there could be only one of the response or there could be more than one responses. So here you can see that again the amplification of signaling has happened, as well as more routes are taken in the signaling process. The third one is here called network type of signaling system, where ligand binding to the receptor now engages more effector molecules which have many more responses, and there could be crisscross of regulation where E1 effector is interregulated by E2, and this E1 is mounting a response through R1, but this response is regulated by the R2 response which was initiated by effector 2. As well as there could be inhibitory feedback signal which is shown here in R3 sending a feedback inhibitory response to E3 effector molecule. Also some of these effectors themselves send the inhibitory signal. So what we see is that multiple different types of signaling nodes exist. Depending on the process, depending on the complexity of the situation being dealt, you can have linear type of signaling system or divergent type or network type. And we have seen plenty of example where all these were coming into picture. Now let's talk about the tyrosine kinases. I have told that this is a kinase which is exclusive to the animal kingdom. There are some animal viruses which actually have molecules which have tyrosine kinase activity, but that is performed only when these viruses infect the animal cells. As we have been seeing any of these kinase activity, it could be receptor type which is of more interest in the signaling, but you have good amount of non-receptor type kinases. In case of plants, serine threonine kinases, you saw receptor-like cytoplasmic kinases. So basically, non-receptor type tyrosine kinases are equivalent to RLCK, which basically will associate with receptor and activate the receptor, or it can individually carry out tyrosine kinase activity independent of receptor. So these tyrosine kinases, or for that matter any kinase we have seen, must have a kinase or catalytic domain which will be ensuring the phosphorylation of that residue happens, and in this case it will be the tyrosine residue which will be phosphorylated. Then you must have that tyrosine residue which is getting modified. So often we will see in coming classes that Y is shown on the protein where phosphorylation will be happening. In addition to these, if you talk about the receptor tyrosine kinase, you have this transmembrane domain, and we have seen this in case of many other receptors, that there is basic minimum 3 domain requirement: one extracellular domain which binds to the ligand, a transmembrane domain, and then you have a cytosolic domain, and this domain has the kinase activity. So for discovery of some of these important molecules which are serving as ligand, EGF, epidermal growth factor, or NGF, nerve growth factor, Stanley Cohen and Rita Montalcini were awarded Nobel Prize in Physiology or Medicine. We will see that how the signaling which is going to be mediated by receptor tyrosine kinase plays very important role in overall development, growth, survival, and differentiation kind of aspects of animals. Let's focus on the discoveries which happened, which has now led to a big area of receptor tyrosine kinase functioning and so on. So one of the earliest discovery for which Stanley Cohen and Rita Montalcini were given Nobel Prize was the first thing in direction of receptor tyrosine kinase or tyrosine kinase signaling. So EGF and NGF were discovered. Then further it was noticed that cancer cells, they produce these factors which are called growth factor, and the specific type of growth factor will affect growth, proliferation and other aspects of a cell of a specific kind. Like nerve growth factor is basically working on neurons. Epidermal growth factors is working on all epidermal type of cells. Then protein tyrosine kinase activity was detected for the first time in this molecule which is called SARC, and this comes from the sarcoma virus. And it was very clearly demonstrated that this has a very specific tyrosine kinase activity. We will see little more of that in some time, where it was very clearly demonstrated that it is tyrosine residue, not any other residue which is getting phosphorylated. Just to put things in place, at the time when these discoveries were being made, serine, threonine residues were known to be phosphorylated. Then further discovery were made that EGF, actually the epidermal growth factor that signals through protein tyrosine kinase, it was still not very clear whether it is a receptor tyrosine kinase type. Then further discoveries as and when the research progressed in this direction suggested that receptor activation requires dimerization. This I have told multiple times, whether we discuss the receptor histidine kinase or receptor serine threonine kinase, that receptor dimerization is one of the earliest event towards the activation of the receptor and signaling to move forward. Then there were specific domains which were identified, which will now become very important when we will discuss the signaling downstream of receptor tyrosine kinase. One of the domains is called SH2 domain. SH is for SARC homology, because these domains were first identified in the SARC kinase which was isolated from sarcoma virus. Another set of domain and their structures were solved. SH2, SH3, these domain structures were determined. Then one interesting aspect was found out, that there are proteins which have tyrosine phosphatase activity. Remember, any signaling which was started had a nice balance, because kinases could add phosphate to a given residue and phosphatases could remove the phosphate, so that equilibrium is maintained. Same thing here was identified by detection of tyrosine phosphatase domain. Further, more and more domains were identified. One of them is called pH domain, which helps in binding with the PIP2 lipid. So some molecules which have to interact with the receptor tyrosine kinase, they are membrane bound. For membrane binding, they have this pH domain which helps them associate with the PIP2 lipid. Then one of the pathways which was detailed downstream of these receptor tyrosine kinase signaling was RAS MAP kinase pathway. I don't need to tell you again about the MAP kinase pathway, but simply it was like MAP3K was activating MAP2K and it was activating MAPK. And what was found in this, that this MAP3K is activated by RAS like molecule, and RAS is a small GTPase. We have discussed this earlier and placed an emphasis that how these small GTPases play very important role in the signaling event. Another domain was identified, PTB domain, which is phosphotyrosine binding domain. The SH2 domain which was found, that is also phosphotyrosine binding domain. A very different arrangement of domain in proteins was found where it was called PTB domain or phosphotyrosine binding domain. We will see more details that how these domains differ when it comes to recognizing the phosphotyrosine residues. Then there were a set of enzymes, PI3 kinase was found to be working downstream of these receptor tyrosine kinase, which activates the AKT pathway. AKT is a protein kinase of serine threonine type. Then further discoveries of receptors for EGF and FGF fibroblast growth factor were found. The structure of this complex of EGF with EGFR was solved, and then there were drugs which were targeting the RTKs were approved for the cancer treatment. Finally, but not the last thing, that another very unique set of receptor tyrosine kinase were discovered where EPH was the receptor, ephrin was the ligand, and this set of EPH ephrin pose a very unique type of receptor tyrosine kinase signaling where receptor and ligand both are membrane bound. Now in some sense this discovery which I listed over there started very early in 1960s, and where we stand today with lot of understanding of the receptor tyrosine kinase in 2020s. And one very striking discovery which was made is around 1980s, and that was the discovery of SRC kinase. I briefly mentioned to you that this was a very important discovery because until then all the kinases which were identified in different systems, they were serine threonine kinase. So it was a landmark discovery, plus what is also called in today's world a disruptive science, which actually brought to the fore a very new kind of activity which was very important in animal kingdom, because as we understand today, different aspects of growth, survival, proliferation, differentiation, and development, early development of tissue is being regulated by different types of RTKs. In today's class further, we will see how some of these were identified and what are their functions. So what I mentioned, that this particular discovery which is made here, what has been shown very clearly in this paper in PNAS, that a new kinase activity has been identified, and that is specific to tyrosine. So the experiments which were performed here is more like TLC and electrophoresis. As we all understand that in TLC you can spot the sample at a particular place, and along with the mobile phase the molecules move into that thin layer material which is up over there. Going by the soluble going by the mobile phase, and the nature of the molecule which is being resolved on this chromatography. There are definite spots where a particular structure, where a particular compound can be detected. So in that field, it was very clearly known that using a given type of soluble, using a given type of mobile phase, if you are starting the sample from here and doing the TLC electrophoresis, you would expect the phosphoserine band at this location and the phosphothreonine here. But repeatedly the scientists found out that there is a band or there is a spot which is coming, which is very distinct from phosphoserine and phosphothreonine. Again, using that same VSARC or the viral SARC protein and performing experiments on different proteins, target proteins. One of them was immunoglobulin molecule, and it was analyzed further. When you spot the sample here, generally what is expected for phosphoserine and phosphothreonine is a spot here. But to their surprise, they did not find any spot over here. Rather, they found spot only at a new place, which they said this is the phosphotyrosine. So different control experiments under these conditions A, B, C, D were performed to establish that the new kinase activity which they are seeing with this SARC kinase is phosphotyrosine activity or tyrosine kinase activity, not the serine threonine kinase activity. And this was such a remarkable study by Tony Hunter, the lead author over there, that everything which is shown, this is a review of their paper, and everything is yes, 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 and so on. And more importantly, the comment which was there, that this is a clear, well written manuscript of great significance, and this describes an unusual specificity for tyrosine residue, and there is important aspect of SARC activity which has been described, SARC kinase activity which has been described, and the reviewers say that no revision is required and immediate publication is strongly recommended. These are the choice of words which are unheard of in today's time of publication. So it was a great study, disruptive study, which established that animals have a very unique type of kinase activity that is towards the tyrosine residue, and SARC became the champion molecule. Now, what we know about the tyrosine kinase activity, the receptors and so on, we all understand that receptors must have minimal property of an extracellular domain, a transmembrane domain, and intracellular domain. If you recollect our discussion on serine threonine kinases type receptors or histidine kinase receptors, we found that how intracellular domain is the one which has kinase domain in it and shows the least variability, and the extracellular domain which binds to the ligand has the highest variability. Similarly, here in case of receptor tyrosine kinases, there are multiple ways by which extracellular domain is arranged. One of them is a split cysteine rich domain. So what basically it would look like, that outside you have a split arrangement of the extracellular domain if I write ED. Then there is a unique example in case of insulin receptor, that the receptor is like two units are there, one which is transmembrane and the other unit which is here is held by disulfide bond. It seems like this is evolving along with the insulin itself, which has two disulfide bond between beta and alpha subunit of the insulin moiety. So one arrangement was where your receptor was outside and it has the split domain arrangement, cysteine residues were enriched. There could be a leucine rich repeat kind of domain which will be present, and not to mention that internally they both have the tyrosine kinase domain. So, other important domain which is seen with the extracellular domain is called immunoglobulin fold domain. So, immunoglobulins are known to having a fold which can be considered like this, simply arranged shown like this, that immunoglobulin fold like domain which is present. So, many of the receptors which are in different families, whether it is epidermal growth factor receptors or so, you have an extracellular domain which has similarity to the immunoglobulin folds. Then there is interesting type of extracellular domain which is called fibronectin type 3 domain, which will be important for receptor to engage in interactions with the extracellular matrix, because this is one of the very critical aspect in some of the kinases which are part of focal adhesion and interact with integrin kind of molecule. So, extracellular domain is something which is very unique, which makes the receptor specific to a ligand type or a class of ligands to which they can bind. In case of receptor tyrosine kinase, there are additional changes which are there in intracellular domain. So, there is a domain called insert domain which is inserted in the kinase domain, which makes it a split kinase domain. So, this is how variability has been imparted in the receptor tyrosine kinases by virtue of changes which is happening in extracellular domain or changes which are happening in the intracellular domain. Now, if we look further, what we will realize that there are multiple of these receptors which are important and they play a great role in signaling, and they are very critical for entire well-being of the organism. So, you have the receptors like NGF that works through this receptor which is called TRK receptor. This is tyrosine receptor kinase type receptor. This is important for the survival and growth of the neurons. Then you have insulin kind of receptors which have been helping cells and tissues for the carbohydrate metabolism and the protein synthesis aspects. Then you have IGF like ligand which works through, this is insulin like growth factor. It will have insulin as growth factor receptor. This is important for survival and growth of many cell type. Then you have EGF, which is epidermal growth factor receptor binding ligand. So, this is the receptor for that. It is important for survival, growth, differentiation and proliferation. So, what you are noticing if you look at this table, that most of these receptors are helping towards survival, growth and differentiation. So, entire aspect of animals survival, growth and differentiation and sometimes the migration of cells for a better cause is dependent on the receptor tyrosine kinase signaling. Then you have fibroblast growth factor and its receptor. The fibroblasts are the cells which are found in the connective tissue, and the entire connective tissue is prepared or generated by the fibroblasts. So, it is important for their survival. It also inhibits the differentiation of some particular type of cell so that they retain their original identity. Then there is something very unique which is called VEGF, vascular endothelial growth factor, which is important for angiogenesis. So, this angiogenesis, particularly if you think about the cancer where cells are rapidly growing and you need more and more nutrients coming from the blood vasculature. So, you will allow more and more angiogenesis to happen. So, thus in many cancer you will see that VEGF are VEGFR is actually working. Then you have something called platelet derived growth factor that will be working through its receptor PDGFR, and this is also important for cell survival, growth, proliferation and migration. This is very unique case where cells need to migrate rapidly, and that is where they will need this platelet derived growth factor. You can understand one of the prime source from where such a factor is coming originally was identified from platelet, which is part of the blood cells, and when blood clotting has to happen, a lot of cells have to move in that direction. That is how this particular signaling works. Then eventually you have a ephrin and EPH. The ephrin is the ligand, EPH is the receptor. This is very important for exon guidance, angiogenesis, tissue boundary formation. We will see how during the development, you know that intestine has distinct regions. What makes sure that there are cells of one type, second type and third type, and the tight boundary which is maintained. And this boundary maintenance is very important for normal development of the tissue. In addition, we have lot of non-receptor tyrosine kinases, which basically are soluble type of kinases. They also have SH2, SH3 domain. Some of them come with the DNA binding domain, like in case of this particular kinase, which is called Abel kinase. Then they also have the pH domain, which is important for the PI-P2 binding. And then you have integrin binding domain, which is present in the FAC kinases. So in general, you have, if you think about the non-receptor tyrosine kinase and look for the domain arrangement, here you are not worried about a domain which will bind to the ligand, because this is intracellular. So one of the domain they can have or they must have is the tyrosine kinase domain. Then they can have in addition another domain which is called SH2 domain, which will help in the binding with tyrosine residue, which is phosphorylated. It will recognize specifically that residue. They can also have additional domain which is called SH3 domain. This will bind to residues which are having a stretch of proline residues together, polyproline sequences to which it binds. In addition, they can have multiple different domains. I already told you that they can have DNA binding domain and activation domain to serve like a transcription factor. They can also have a theoretically phosphatase domain. So we'll see in coming classes that how lot of these molecules which are critically required for tyrosine kinase signaling, they come together and give us different types of signaling downstream of receptor tyrosine kinase, like linear, divergent or network type. So in essence, what we have understood in this class today is that tyrosine kinases are soluble and receptor type, no exception. We have seen histidine kinases also of that sort, serine and threonine kinase also of that type. Then these are important for growth, proliferation, differentiation and survival. There are conserved domain which are present in both receptor tyrosine kinase and non-receptor tyrosine kinase, like SH2, SH3 along with the kinase domain. And then there is a huge diversity which will be which we will see downstream of this receptor because of multiple adapter and effector like molecule which will respond to the kinase activity which generates the phosphotyrosine residue. Thank you.